It's a great honor to be here, and I kind of, want to take you through the journey, which is this discovery of the cells that make up the human body. The first effort is to actually say, what is that variety of cells that we have? And so this initiative that Kat mentioned was, kick-started here in this building in late 2016, which was a global effort to map the human body, and it was called the Human Cell Atlas Initiative, a reference atlas of what are the cells that make up the human body, and then if you look at disease, you can then say, what are the variations that you see in the global diverse population, and what goes wrong in disease? How is the gene expression altered? So this is to create a reference atlas of human cells. So what did we discover? And I'm going to take you through some of the vignettes. When it first started, it was just before the pandemic. And then the pandemic hit. And the first thing that you could do with the data set that was already there from multiple organs was to actually look at which cells are expressing the receptor that allows the virus to enter the cell and cause the infection. So all of us who were working on the Human Cell Atlas pulled together data that we hadn't even published to ask this question and identified that the viral entry receptors were found in the cornea, conjunctiva, and the nasal cavity, and the oral cavity as well. So this really gave you the evidence of why you need to wear masks and also eye protection for some of the invasive procedures that were happening in hospital. So this is the value of such data set and how it can be marshaled and used for global benefit. So coming back to how the strategy of atlasing, and essentially we divided the human body as well as looking at development, pediatric, and genetic diversity into 18 biological networks that characterize and focus on these components so that we can then bring all of that information together. And my contribution has mainly been in development and the skin. So I'm going to just illustrate with some vignettes. So the developmental cell atlas was something that I felt very passionate about because a lot was known about newborn and children, but very little was known what happens before birth in the prenatal stages, because it's very difficult to get the material and to study this. But essentially what happens in utero sets a lot of things in motion after birth. And what we were very lucky in the UK to have was this human developmental biology resource, which was based in London and actually also in Newcastle, very close to my laboratory. And I thought this was a fantastic opportunity to really pioneer research, and hence the Developmental Cell Atlas arose as part of the Human Cell Atlas Initiative. But it's just to emphasize that this resource has been funded by Wellcome and MRC for more than 20 years. And it's having these resources that one can tap into, really understanding the value of what they can bring. And because it's already come with the ethics and also the kind of like research principles that you can adhere to ensure that this is done in a way that is in accordance with standard community practice and beneficial. So being kind of like primarily trained as an immunologist, I thought we should look at early life immunity as a way of starting the developmental cell atlas work. And essentially, we looked at multiple organs from the same embryo and fetuses to work out how the immune system develops from the organs where they are made to the organs where they mature, and subsequently to the peripheral networks, for example, the skin, the gut, and the kidney, where they reside and form your immune network surveillance system. And we published this over several years with each manuscript focusing on one organ. But it isn't just the kind of like large scale, let's do it on every organ. This was carefully considered because these were the organs that were relevant. And this brings and spans 3 disciplines at least in a way that you can't do before, which is hematopoiesis, which is the study of how blood and immune cells form, immunology, the study of the immune system, and developmental biology, which is the study of human development. So here, in one effort, we've got an amazing ability to look across disciplines and to really drive biological insights. And it allowed us to put the cells in space in terms of where they are and across time, i.e. gestational time. And if you now start to look across lifespan, we can begin to sort of gain insights into how we age and how we can actually promote healthy aging or health span. So this was a huge sort of personal kind of excitement on my part, and I think the data sets are very valuable and used by a lot of people. But the ability to do this at scale and to look at multiple cells, multiple organs all together, really allowed you to look at the immune system and see how the whole of it is greater than the sum of its parts, and begin to appreciate some of the emergent properties that you won't see. And I've tried to illustrate with this kind of bird murmuration pattern, which you won't perceive unless you actually study a flock of birds, but you won't see if you study very few birds. So this is the power of such data sets, and this is the power of such initiatives. Kat talked about the big splash, and we had a collection that was published by the Nature Portfolio journal group, which is towards a first draft of the atlas, and just some vignettes from there. Here, remember the 18 biological networks. So we took some of the data sets from all of the different biological networks and integrated them. By that, I mean you will have different data sets that cover different populations and different cohorts, so you can now bring them together and create a reference atlas for that organ that everyone else can use. So this gives you a consensus cell atlas from multiple studies. Very, very important in terms of ensuring the relevance to a broader population. It's always very important when you want to do something like this that there is accompanying data science and computational tools that are involved in actually assembling all of those data sets together and making sense of that data. And we were very lucky from the onset. The Human Cell Atlas, the two co-chairs, Sarah Teichmann and Aviv Regev, were both computational biologists. And so this was always at the heart. And they brought along computer science, data science, really rooted in computer science, and more recently, a lot more of artificial intelligence, and gearing towards how do we make sense of these large data sets? How do we actually start to see the emergent properties? How do we predict? And how do we impute? And how do we generate missing data? All of that are very exciting things to look forward in the future. And coming back to the third component that was featured in this publication collection was the updates on the developmental cell atlas. And I just want to illustrate that it isn't just about a catalog of cells, but it's also what can we learn from that. And I'm going to illustrate with two examples. So here is one from my own group, which is looking at the prenatal skin. And what we found when we actually did the analysis was unique properties of the prenatal skin that was very different if you cultured skin in a dish, which is what we call organoid, and that the presence of immune cells actually helped formation of blood vessels in the skin. So this tells you that immune cells are not only playing a role in terms of protecting us against infection after birth, but in very early life, playing a very important role in how the organs and tissues are formed. So these are the kind of like biological insights that you can extract. And in this amazing video is how the human cartilage and skeleton forms within the developing embryo, where it begins, where they are, and the network that they form, which you can't quite perceive, especially if you were to kind of like break apart an tissue and just have a list, a catalog of cells that make up a particular organ. So these are the kind of like added information that you can glean and really informative about the biological systems that are being studied. So where next for the HCA? Short term, I think we need to democratize this data. What does that mean? Yes, we have all of the data available to be accessed in public repositories, but essentially it's not of any use to most people unless you know how to code, you have data science expertise, or you work in a place that has the computing infrastructure that enables you to analyze large data sets. there's a huge hidden potential that can be unlocked. And what we need are essentially a bit like the apps on our smartphones that allows us to shop at Amazon or IKEA or wherever else. And so having a web portal for the Human Cell Atlas data, which is equivalent to the ensemble, for example, for the Human Genome Project. The value of the Human Genome Project data was really unleashed by having ensemble, so people can access it and essentially use it. In the same way that AlphaFold has been hugely valuable and beneficial to everyone because there are AlphaFold web browsers that can be used. And this is what's needed, because otherwise very few people will benefit from it. How do we get from a vision, for example, the Human Cell Atlas, to actually kind of having all of these successful outputs and building the community? And here I want to kind of like reflect on what we've learned, lessons from the Human Cell Atlas initiative in terms of research culture and infrastructure. The first is the link between vision and culture. It is intimate. It's essentially two sides of the same coin. And even if you had a great vision, if you didn't have the right culture, you'll never achieve the vision. And this is something that I passionately believe in. And it was very useful for us that from the very onset, that culture within the Human Cell Atlas was very much inclusive and supportive and collaborative. And that allowed all of these successes from the HCA to be derived. The second, I want to focus a little bit because it's very different to do a global initiative like this in terms of what is needed and what enables it compared to the kind of like conventional types of research, which is very much driven by individual group leaders. You need to have the right infrastructure, the framework. How do you work together, team science? How do you adapt the technologies? And how do you share knowledge across the group? And you have to balance this with the individual's careers as well, and the publication credit, because these are very important in terms of how do you judge somebody's success. How do you ensure that junior staff are trained accordingly rather than sort of just generating data for a broader initiative and their career becomes subsumed within this initiative? And how do you go from data to understanding biology and mechanism? So these are really important things that we have to balance and actually allow both to kind of like flourish at the same time. And this was from the very start keen that it was very inclusive and diverse so that it will be of global benefit for everyone in terms of health and disease. And I'm not going to read out all of the statistics, but this is very much a global effort, and it's fantastic to see the engagement from around the world. And as I speak, the Human Cell Atlas Asia meeting is happening, which is just amazing to see the kind of like momentum that's going outside of the UK and the global north and the North European and American community. So I talked about the importance of diversity, equity, and inclusion, and as well as ethics. So the Human Cell Atlas really from the very onset had this as part of its ambition. You can see the number of countries where we've got researchers contributing to the Atlasing initiative, as well as number of researchers. And part of this is to kind of have a framework of what it means in terms of how does the global community benefit from atlasing and these types of data sets, but also the logistics of undertaking these types of initiatives, because you have ethical issues and equity issues that need to be addressed. How do you share data? And part of the Human Cell Atlas was to set up an equity working group and also an ethics working group so that the learnings from those groups and the Human Cell Atlas itself can inform the future direction of research where we do want global participation. And as always, you can generate a catalog of cell types. This gives you an enormous amount of data, which is very valuable resource. But what is the knowledge that is buried in that data, and what does it mean? And this was always very important for me particularly, but also for the Human Cell Atlas community. I'm going to end on my personal journey as a clinician scientist, which some people have published and called as the endangered species. My journey started many years ago, and the first time I received Wellcome funding was back in 2009, and I was I think 8 to 10 weeks after I'd given birth. And you usually take the train well in advance so that you're there on time in case the train is delayed. And 8 to 10 weeks after birth, I remember sleepless nights, breastfeeding, and emotional turmoil. And I was thinking on the train journey, what am I doing? I've left my little child. I'm on this train going for an interview. And I went to collection to spend time before the interview, and there was this amazing exhibition called Bobby Baker's Diary Drawings, and it charted the journey of Bobby Baker, the artist, through her time through mental illness and as well as breast cancer and coming out of it. She drew every day of her life, and somehow going through that exhibition, I came out at the other end feeling this. I know exactly what I want. It was such an uplifting exhibition, and it set me in the right frame. So I do believe in the power of art and what collection does in terms of how it can affect individuals. And it certainly affected me because I wouldn't be here otherwise. The second is with regards to having this dual identity as a doctor and a scientist. And I don't know if you've watched this film. Carol Morley happened to be one of the Wellcome Screenwriting Fellowship awardee, and this Screenwriting Fellowship has produced amazing directors. And she basically made this film based on the collection that was from Audrey Amis's family after she passed away, which was kept, that collection, and it inspired her to make this film. And typist, artist, pirate, king is what Audrey Amis stated as her occupation on her passport, which I thought was really a bit like, sometimes I do think, am I a doctor? Am I a scientist? What am I? But the value of being bilingual across two fields is really important, and you can just think about how it is if you're bilingual across two cultures, and how seamless the interactions are and the things that you can perceive. But very much like in this film, this is a journey that Audrey Amis, the one with the glasses, takes from London to Sunderland, where she comes from, accompanied by her carer, who very calmly takes her through this journey. And in many ways, this is how I felt with all of the Wellcome Discovery Research team staff who have held my hand over the years and taken me through that journey. And so, thank you.
Easy.
Yeah, it sounds like a very easy question. It's a bit like, in the Human Genome Project, they had a bet, actually, about how many genes there were, and people were far off. But if you ask that question now, and as I say, the numbers are varying, doubling, even, up to 5 times more than what is thought. So I think this is a bit of a question of having the resolution of how do you kind of like lock to say there is no more cell types? How long is a piece of string at this point? It's a little bit, there. But I think fundamentally it's not necessarily about how many cell types. It's basically having a usable reference map of cells that is going to benefit everyone in the way that knowing the DNA sequence, even though when it was announced as completed, it wasn't complete because it wasn't sequenced from end to end. But whatever there was very valuable and very helpful in terms of enabling people to sort of like see what was normal variation and what were the variants that predisposed to disease. The other way to kind of like look at that question is how much money we have to spend on that. And if you look at the Human Genome Project, that was billions. We haven't even hit there with the Human Cell Atlas. So, I've thought about that long and hard, being a minority myself. And in many ways, if I reflect on myself, I think, I've had to learn how to navigate in a completely different culture. I came to the UK to medical school. I did my A-level equivalent in a different language in Malaysia. And you learn how to navigate. And I think for me now where I am, I kind of like want it to be easier, and I kind of like want to tell people how to navigate. But also to ensure that this doesn't mean that you lose your identity in that process, that you just become what you're supposed to be in order to be successful. So part of that, I think, is how do you even get more diversity into, for example, biomedical research? And a lot of the time I feel it's about how you assess individuals. Often it's about achievements, but for me the achievements are relative to the opportunities that people have had. And we don't pay enough attention to kind of like what has somebody achieved relative to the opportunities that they've had. And therefore, what you end up having are people going into PhDs after they've had several rounds of masters, etc., and they tick everything. But it's because they could afford to actually do all of those, have the working experiences and whatever not. And I really start, and I emphasize this, and I do that with my own recruitment, looking at achievements based on opportunity. And I think that actually reduces the barrier for people to enter. And then actually being more aware of the way different people kind of like, the different cultures, etc., and, you know, really kind of like having that understanding and wanting to learn. Currently, as the interim head of program, I try to make sure that I say, you know, I wish everyone the different kind of like festivities and celebration. It's a small gesture, but it means that people feel they are seen, they are heard. You know, just kind of like asking somebody how they're feeling, if there's a global kind of like event that affects them. And if you're not, you don't feel seen, you don't feel heard, you don't feel you belong, and therefore there's attrition. So there are multiple prongs. I think achievement based on opportunities is one. Making people feel that they are seen and heard is another. I'll address the first one. So in terms of technologies, I kind of tried to allude to the fact that we're now looking at 2-dimension. And I think where it will be is where genomics meets imaging, because both are becoming blurred. Imaging has always been slightly separate to genomics, because genomics has always been about DNA sequencing, but with RNA sequencing and being able to sequence in space, being able to kind of like look at, you know, sequencing data across larger volumes is going to be important. So I think imaging and genomics and how they interface is going to be a really exciting area to explore. So imagine an MRI scan or something, but what you now have are basically gene expression. That's probably what the future is going to be. So we need to understand, you know, and have experts from both fields. Machine learning and artificial intelligence has become extremely, extremely attractive. And many people outside of academia are paying a huge amount of money, and this becomes a real issue in terms of how we attract them. But we've also got to start thinking about how do we actually work, you know, and bring down these kind of like traditional barriers of how you work between academia and biotech, or academia and, you know, industry and so on. So there's a bit of work to be done in terms of how does it benefit, rather than we all generate the data and you all kind of like make the most out of it. How do we actually work together so that everybody, you know, moves at pace? And also it's very important that anything that comes out of the data or how we attract those people, that it is for the global benefit and not for the select few.
I wanted to touch as well on the endangered species kind of aspect of the what we call the kind of dual career, where you're active in the clinic, you're seeing patients, you know, you have specialism in dermatology, and then you're also doing this kind of discovery research in the lab. And I wondered if you could expand a little bit on both the challenges of that in terms of kind of flipping across the modes or the cultures or the languages, but maybe also some of the inspiration that you draw.
I'll start with the inspiration first. So for me, I knew early on that I enjoyed research. I liked the flexibility, but I also felt, first and foremost, I was a doctor. and the clinical work grounds me. So the motivation might be different for different people, but for me, it's kind of like reminding myself why I'm doing the research. And also, I often find that when I see my patients, I see a problem in a completely different way. So there's always this, really helpful dialogue that grounds me and also gives me inspiration. In terms of challenges, it is challenging. It has become more challenging. And there are several reasons, not necessarily because anybody is against clinician scientists, but it's just, some things that change and evolve. I benefited from Wellcome having specific clinical fellowships. So I went through that scheme, and I was really lucky to kind of go from an intermediate clinical fellow to then a senior fellow, and which I am still now. It's now been combined, and there are strengths and weaknesses to it. So that's something to kind of consider. The other thing is as well with regards to how you actually, the incentives for you to do research as a clinician, and also how you enter into the research scheme. So one of the initiatives that were put forward was to kind of like have this academic route. So people who are very interested in doing to become a clinician scientist could get into that. But the problem meant that unless you got into it, it was harder for you to get into it at a later stage. So that, inadvertently created barriers, so made it less inclusive. And these are things to consider. And the third thing I want to touch upon is this kind of like valley of death, as I see it, basically between PhD and the next stage. There are very few schemes that support individuals at that stage. And the way that we have structured the stages means that it particularly is bad for people who train part-time. And a lot of women who train part-time because of young children actually have a longer period of time when they have to do their specialty training. And as a result, by the time they qualify for their next stage, they've spent many years without doing research, and therefore are seen as less competitive for when they apply for their next stage. So there are several areas that I think where we can capture and try to promote this. In terms of trying to get more people who come in from outside of the ACF, I've tried to do that as part of my role within Newcastle Biomedical Research Centre, the NIHR-BRC, really trying to kind of like showcase individuals who've not gone through the conventional track, and giving opportunities like, startup, like a pilot 1-year funding so that they can, become competitive to get their clinical research training fellowship. And for the next stage, I think I've tried to campaign via the Academy of Medical Sciences that they're the only people giving those schemes, the fellowship schemes. So we need to do something about that. And the final point is, how do you make it easy for people? And it's also dependent on specialty. So surgical specialties or trade specialties. And the one advantage of the clinical fellowships meant that you were primarily there to do research, and your role in the clinic is to support your research. And that was why I benefited from that, because it enabled me to spend 80% of my time doing research and 20% of my time doing clinic. But if you didn't have that, it becomes 50%, 50%. And to be honest with you, it's almost impossible for me to do, because nothing is really 50%, 50%, with clinical work. It would have been impossible to juggle with little children and everything else. So there's a lot of disincentives without this kind of like, protection from the fellowships.